All right, welcome back. This is part 2 of the week 7 lecture. Again, this is a reading heavy section where we're in chapters 4 and 5 of the Concepts of Biology textbook. We're continuing on in chapter 4 talking about glycolysis, the citric acid cycle, and oxidative phosphorylation. These are three main key points or key biochemical pathways whereby organisms are able to utilize the sugars that they consume to gain huge amounts of energy by the end of this process. So we start out with glycolysis, and if you're following along with your with the book in front of you, as you're looking at this, the figures associated with glycolysis. So this is figure 4.13 in the textbook. Again, Concepts of Biology textbook. We're starting out with this 6-carbon ring. That's glucose. And for me, when I think about these processes, I like to follow the carbons through this process so I can understand where this energy is coming from. This process of glycolysis occurs in the cytoplasm of all cells. Whether it's a simple bacteria, some bacteria only use glycolysis as their energy harvesting sort of mechanism. Humans and plants are more complex and need more energy, so they use glycolysis is the first step in this process, but they have additional steps where they're utilizing mitochondria, these organelles inside eukaryotic cells, to get more energy from these glucose molecules. So we're starting out with these glucose molecules. To break them apart, we start out and we invest 2 molecules of ATP, or adenosine triphosphate. This is an energy investment step in this process. It breaks this glucose molecule in half and turns them into phosphorylated or high energy 3-carbon sugars. You can see on this diagram that there are these sort of long arrows that point down towards these molecules called pyruvate, and understand that these arrows are really just representations that these are biochemical pathways driven by enzymes to change these molecules' shape and form. So we start with the 6-carbon glucose molecule, and at the end we end up with 2 3-carbon pyruvate molecules. Additionally, we've gained a little bit of ATP. If you look at the bottom of that diagram, you see there's 2 ATPs coming off those little arrows. So we have a total gain of 4 ATPs, but we've invested 2 at that first step. So with glycolysis, we get 2 molecules of ATP for every molecule of glucose. Now, 2 molecules of ATP is not very much. Cells can't do very much with only 2 molecules of ATP, and that's why only simple bacteria can really utilize glycolysis as their only metabolic pathway. Eukaryotic cells like plants need more energy, and so we have more advanced ways to continue to break down these glucose molecules and harvest more energy from the potential energy stored in those carbon-carbon bonds. It's important also to understand that this glucose, the citric acid cycle, and oxidative phosphorylation are all parts of a big, long energy movement train. We break it into these parts because they're easier to manage and understand, but you also need to understand when we're talking about oxidative phosphorylation and full use of glucose molecules for energy, that these are all part of a bigger chemical process that we use to harvest energy more efficiently. And by we, I mean eukaryotic organisms. So the next step in this chain is what's called the citric acid cycle. The citric acid cycle starts, again, now we're looking at figure 4.14 in the text. So we're starting with pyruvic acid, and these are the pyruvates from glycolysis that have been changed in another step that we don't really talk about in this class. But understand that this 3-carbon pyruvic acid molecule is the pyruvate from the previous end product of glycolysis. So this pyruvic acid, these pyruvate molecules are moved from the cytoplasm of the cell into the inner space of the mitochondria. In this inner space of the mitochondria is where we have the citric acid cycle. And again, let's follow the carbons through. So we have this 3-carbon pyruvic acid molecule inside this, inside the mitochondria. The first step is this coenzyme A breaks off one of these carbon molecules and it off-gasses as CO2. The CO2 that you're breathing out, that's where the CO2 is coming from. It's a waste product or a byproduct of this chemical reaction. So we've went from a 3-carbon molecule and we've lost one of them, and so now we're down to a 2-carbon molecule called acetyl-CoA. So we have 2 carbons attached with this coenzyme A. It then moves into this other cycle called the citric acid cycle, where the coenzyme A is removed and these carbon bonds are broken. So we've got 2 carbon bonds left, or 2 carbons bonded together left, and they break apart into 2 individual molecules of CO2. So we start with a 3-carbon pyruvic acid. The first carbon is broken off as a carbon dioxide, and then the final 2 are then split into 2 more molecules of carbon dioxide. So again, this is where the carbon dioxide that we breathe out comes from. Plants also exude carbon dioxide through their leaves as part of their metabolic processes too. That's an important thing to understand, that plants definitely, I don't want to say breathe, but respire out this carbon dioxide. They just also happen to produce an abundance of oxygen, more so than they need, and that's why we have the oxygen that we need in the atmosphere. At any rate, so this 3-carbon pyruvic acid is broken apart into 3 molecules of CO2 during the citric acid cycle. There's also a little bit of gain of ATP as part of the citric acid cycle, but again, it's only 1 molecule, so almost negligible amounts. What we really get out of the citric acid cycle are these 2 molecules called NADH and FADH2. You can see at the top of the circle there, the citric acid cycle, we have this conversion of NAD plus to NADH and FAD to FADH2. The NADH and the FADH2 are really the important molecules when we're talking about the citric acid cycle, because these are the high energy molecules that move into the electron transport chain and oxidative phosphorylation in the next step along the line. This is these molecules are a way, when these carbon bonds are broken from, when we're breaking apart the pyruvic acid, the building of the NADH and the FADH2 are how biological organisms are reassigning that energy from the carbon-carbon bonds and putting them into these different chemical bonds. And the NADH and the FADH2 have what are called high-energy electrons built into these chemical bonds, and those high-energy electrons are what are going to be used moving forward into the next step along the electron transport chain. Moving into oxidative phosphorylation, we've got these 2 main components to the oxidative phosphorylation parts. We have the electron transport chain and the ATP synthase components. I think it's really critical to understand that this part of this energy building process is our physical structures. The electron transport chain and ATP synthase are things. They are very small things. They are proteins embedded in the membrane of the mitochondria, but they are things. And I think that's a useful way to think about it. And these things are essentially little tiny pumps. These little tiny pumps are powered by these high energy electrons coming into the system via the NADH and the FADH2 molecules. The NADH and the FADH2 molecules are oxidized, hence oxidative phosphorylation. They're oxidized, and when they're oxidized, they release their high energy electrons. And these high energy electrons are then put to work in this sequence of proteins, and these high energy electrons are what are powering these tiny pumps. And what these tiny pumps are pumping are hydrogen ions, protons. They're moving protons through the membrane, the inner membrane of these mitochondria into the inner membrane space. You have to refer back to a previous chapter looking at the structure of cells. Please do so and look at the structure of mitochondria and put it into the context of this, that mitochondria have an inner and outer membrane. The electron transport chain in this case is occurring in the inner membrane of the mitochondria, and it's pumping hydrogen ions into this inner membrane space. What pumping of hydrogen ions does is it sets up what's called a chemiosmotic gradient. It's essentially a diffusional gradient where there's more hydrogen ions on the upper side or the inside of this inner membrane space than there is on the inside of the intermembrane space. Setting up this concentration gradient on either side of this membrane makes these hydrogen ions want to move back across the membrane to even out the concentration gradient on either side. When these hydrogen ions move back through the membrane, they do so through this complex called ATP synthase. ATP synthase is a protein complex that utilizes this energy from these hydrogen ions diffusing through the gradient to attach phosphates to ADP. Again, this is a physical structure that's putting these molecules together. This physical structure embedded in the inner membrane of the mitochondria is doing this work, and it's utilizing the energy of this chemiosmotic gradient, moving these hydrogen ions from one side to the other and picking up a little bit of energy along the way and putting that energy to work to add phosphates to ADP to build ATP. Again, so we're starting with the products of the citric acid cycle, the NADH and the FADH2. Those two molecules are donating their high energy electrons to the electron transport chain, these little tiny chemical pumps. That energy from those electrons is pumping hydrogen ions against the concentration gradient into the intermembrane space of the mitochondria. Diffusional gradient, this chemiosmotic gradient, wants to push these hydrogen ions back through the membrane, and when they go through the membrane, they do so through the ATP synthase. The ATP synthase uses those protons moving through to drive this attachment of phosphates to ADP to produce ATP. This process allows for the full breakdown of the six carbon glucose molecules. So we broke it apart in glycolysis, right, into 3 individual pyruvates. Inside the mitochondria, the citric acid cycle further breaks apart these 3 carbon pyruvates into 6 individual molecules of carbon dioxide, but it uses the energy in those bonds to build NADH and FADH2. NADH and FADH2 transfer that energy via those high energy electrons to the electron transport chain, where that energy is put to work in pumping hydrogen ions. That the energy put to work pumping those hydrogen ions sets up that chemiosmotic gradient. That chemiosmotic gradient drives ATP synthesis via ATP synthase, this complex of proteins. That's how we break down glucose fully, rather than this partial breakdown that we get with just glycolysis. And that full breakdown of glucose produces many, many times more energy per glucose molecule than you get with just straight glycolysis. Without the additional energy of oxidative phosphorylation, complex life on this planet would not exist. But it happens in the electron transport chain as part of a physical structure utilizing these high energy molecules. All right, look for part 3, where I'm going to talk about photosynthesis, and talk at you later.